Hello, and welcome to today's reading of Biological Psychology, 13th edition, Nerve Cells and Nerve Impulses, Chapter 1. People talk about growing into adulthood and becoming independent, but in fact, almost no human life is truly independent. How often do you hunt for your own meat and cook on a fire you made from scratch? Do you grow your own vegetables? Could you build your own house with tools you made yourself? Have you ever made your own clothing with materials you gathered in the wild? Of all the necessities for your survival, which ones, if any, could you do completely on your own, other than breathe? People can do enormous amount together, but very little by themselves. The cells of your nervous system are like that too. Together they accomplish amazing things, but one cell by itself is helpless. We begin our study of the nervous system by examining single cells. Later we examine how they act together. Module 1.1 The cells of the nervous system. No doubt you think of yourself as an individual. You don't think of your mental experience as being composed of pieces, but it is. Your experiences depend on the activity of a huge number of separate but interconnected cells. To understand the nervous system, the place to begin is to examine the cells that compose it. The nervous system contains two types of cells, neurons and glia. Neurons receive information and transmit it to other cells. Glia serve many functions that are difficult to summarize, and we shall defer that for a discussion later in this module. The adult human brain contains approximately 86 billion neurons on average. The exact number varies from person to person. Now we take it for granted that the brain is composed of individual cells, but the idea was in doubt as recently as the early 1900s. Until then, the best microscopic views revealed little details about the brain. Observers noted long, thin fibers between one cell body and another, but they could not see whether a fiber merged into the next cell or stopped before it. In the late 1800s, Santiago Ramon y Cajal used a newly developed staining techniques to show that a small gap separates the tip of a neuron's fiber from the surface of the next neuron. The brain, like the rest of the body, contains and consists of individual cells. Figure 1.1 Estimated numbers of neurons in humans. Cerebral cortex, 16 billion neurons. The rest of the brain, 1 billion. The cerebellum, 69 billion neurons, and the spinal cord, 1 billion neurons. Santiago Ramon y Cajal, a pioneer of neuroscience. Two neuroscientists of the late 1800s and early 1900s are widely recognized as the main founders of neuroscience. Charles Sherrington, whom we shall discuss in Chapter 2, and the Spanish investigator Santiago Ramon y Cajal, 1852 to 1934. Cajal's early education did not progress smoothly. At one point, he was imprisoned in a solitary cell, limited to one meal a day, and taken out for public floggings at the age of 10, for the crime of not paying attention during his Latin class. Cajal wanted to become an artist, but his father insisted that he study medicine as a safer way to make a living. He managed to combine the two fields by becoming an outstanding anatomical researcher and illustrator. His detailed drawings of the nervous system are still considered definitive today. Before the late 1800s, microscopy revealed few details about the nervous system. Then the Italian investigator Camillo Golgi found a way to stain nerve cells with silver salts. This method, which completely stained some cells without affecting others at all, enabled researchers to examine the structure of a single cell. Cajal used Golgi's methods but applied them to infant brains, in which the cells are smaller and therefore easier to examine on a single slide. Cajal's research demonstrated that nerve cells remain separate instead of merging into one another. Oddly, when Cajal and Golgi shared the 1906 Nobel Prize for Physiology and Medicine, they used the acceptance lectures to defend contradictory positions. In spite of Cajal's evidence, which had persuaded almost everybody else, Golgi clung on to the theory that all nerve cells merge directly into one another. Philosophically, we see the appeal of the old idea that neurons merge. We describe our experience as undivided, not the sum of separate parts, so it seems right that all cells in the brain might be joined together as one unit. The structures of an animal cell. Figure 1.2 illustrates a neuron from the cerebellum of a mouse, magnified enormously, of course. The neurons have much in common with the rest of the body cells. The surface of the cell is the membrane, or plasma membrane, a structure that separates the inside of the cell from the outside environment. Most chemicals cannot cross the membrane, but protein channels in the membrane permit a controlled flow of water, oxygen, sodium, potassium, calcium, chloride, and other important chemicals. Except for mammalian red blood cells, all animal cells have a nucleus, the structure that contains the chromosomes. A mitochondrion, plural, mitochondria, is the structure that performs metabolic activities, providing the energy that the cell uses for all activities. Mitochondria have genes separate from those in the nucleus of the cell, and the mitochondria different from one another genetically. People with overactive mitochondria tend to burn their fuel rapidly and overheat, even in cool environments. People whose mitochondria are less active than normal are predisposed to depression and pains. Mutated mitochondrial genes are a possible cause for autism, and it cites a study there. Ribosomes are the sites within a cell that synthesize new protein molecules. Proteins provide building materials for the cell to facilitate chemical reactions. Some ribosomes float freely within the cell, but others are attached to the endoplasmic reticulum, a network of thin tubes that transport newly synthesized proteins to other locations. The structure of a neuron. The most distinctive feature of neurons is their shape, which varies enormously from one neuron to another. Unlike other body cells, neurons have long branching extensions. All neurons include a stoma, cell body, and also have dendrites, an axon, and presynaptic terminals. The tiniest neurons lack axons, and some well-defined dendrites. Contrast the motor neuron in Figure 1.4 and a sensory neuron in Figure 1.5. A motor neuron with its soma in the spinal cord receives excitations for its dendrites and conducts impulses along its axon to a muscle. A sensory neuron is specialized at one end to be highly sensitive to a particular type of stimulation, such as light, sound, or touch. The sensory neuron, shown in Figure 1.5, conducts touch information from the skin to the spinal cord. Tiny branches lead directly from the receptors into the axon, and the cell's soma is located on a little stalk off the main trunk. Dendrites are branching fibers that get narrower near their ends. The term dendrite comes from a Greek word meaning tree. A dendrite branches are like a tree. The dendrite's surface is lined with specialized synaptic receptors, at which dendrite receives information from other neurons. The greater the surface area of a dendrite, the more information it can receive. Many dendrites contain dendritic spines, short outgrowths that increase the surface area available for synapses. The cell body or soma, Greek for body, plural somata, contains the nucleus, ribosomes, and mitochondria. Most of a neuron's metabolic work occurs here. The cell bodies of neurons range in diameter from 0.005 millimeters to 0.1 millimeters in mammal and up to a millimeter in certain invertebrates. In many neurons, the cell body is like the dendrites, covered with synapses on its surface. The axon is a thin fiber of constant diameter, rather. I was about to say, what on earth is contrast diameter? The axon is a thin fiber of constant diameter. The term axon comes from the Greek word meaning axis. The axon conveys an impulse towards other neurons, an organ, or a muscle. Axons can be more than 1 meter in length, as the case of the axon from your spinal cord to your feet. George Excoli, 2015, offers the analogy that if you could expand the dendrite of a reasonably typical neuron to the height of a tree, the cell's axons and its branches would extend for more than 25 city blocks. Many vertebrate axons are covered with an insulating material called a myelin sheath, with interruptions known as the nodes of Ranvier. Invertebrate axons do not have myelin sheaths. Although a neuron can have many dendrites, it can only have one axon, but the axon may have branches. The end of each branch has a swelling, called the presynaptic terminal, also known as the end bulb or the bolt button. At that point, the axon releases chemicals that cross through the junction between the neuron and the other cell. Other terms associated with neurons are afferent, efferent, and intrinsic. An afferent axon brings information into a structure. An efferent axon carries information away from a structure. Every sensory neuron has an afferent to the rest of the nervous system, and every motor neuron has an efferent from the nervous system. Within the nervous system, a given neuron is an efferent from one structure and an afferent to another. You can remember that efferent starts with E as an exit, and A for afferent starts with A as in admit. If a cell's dendrites and axon are entirely contained within a single structure, the cell is an interneuron or intrinsic neuron of that structure. For example, an intrinsic neuron of the thalamus has an axon and all its dendrites are within the thalamus. Variance among neurons. Neurons vary enormously in size, shape, and function. The shape of a neuron determines its connections with other cells and therefore determines its function. For example, the widely branching dendrites of a Purkinje cell in the cerebellum, see figure 1.8a, enable it to receive input from up to 200,000 other neurons. By contrast, the bipolar neurons in the retina, see figure 1.8d, only have short branches, and some receive input from as few as 2 other cells. Glia. Glia or neuroglia are the other components of the nervous system, performing many functions. See figure 1.9. The term glia is derived from the Greek word meaning glue. It reflects the early investigators' idea that glia were like glue and held the neurons together. Although that concept is obsolete, the term remains. Glia outnumber neurons in the cerebral cortex, but the neurons outnumber glia in several other brain areas, especially in the cerebellum. Overall, the numbers are almost equal. The brain has several types of glia. The star-shaped astrocytes wrap around the synapses of functionally related axons, as shown in figure 1.10. By surrounding connections between neurons, an astrocyte shields it from chemicals circulating in the surround. Also, by taking up the ions transmitters released by axons and then releasing them back, an astrocyte helps synchronize closely related neurons, enabling the axons to send messages in waves. Astrocytes are therefore important for generating rhythms, such as the rhythm of breathing. Astrocytes dilate blood vessels to bring more nutrients to the brain areas that have heightened activity. A possible role in information processing is also likely, but less certain. According to the popular hypothesis known as the tripartite synapse, the tip of an axon releases chemicals that cause the neighboring astrocyte to release chemicals of its own, thus magnifying or modifying the message to the next neuron. This process is a possible contributor to learning and memory. In some brain areas, astrocytes also respond to hormones and thereby influence neurons. In short, Astrocytes are active partners of neurons in many ways. Tiny cells called microglia act as part of the immune system, removing funguses and viruses from the brain. They proliferate after brain damage, removing dead or damaged neurons. They also contribute to learning by removing the weaker synapses. Oligodendrocytes in the brain and spinal cords, and swan cells in the periphery of the body build the myelin sheaths that surround and insulate certain vertebrate axons. They also supply an axon with nutrients necessary for proper functioning. Radial glia guide the migration of neurons and their axons and dendrites during embryonic development. When the embryological development finishes, most radial glia differentiate into neurons, and a smaller differentiate into astrocytes and oligodendrocytes.